what sustains cancer, why does cancer occur, what sustains it, what can we do to end it. And so we are very pleased to have Dr. Xiaolong Ming offer what I believe will be a kind of bringing together of many of the diverse comments that we made yesterday. I kind of sense a consensus occurring, and let's see if what Dr. Ming has to say, if that doesn't kind of bring additional weight to what we were talking about. So with that, I'd like to introduce Dr. Xiaolong Ming.
Thank you, Dr. Ron. Good morning.
Good morning.
My presentation today, the topic is a new frontier of IBC cancer research. There's many sayings in our lab. One of them is that any investigator who makes a pointed effort to find a cancer cure is likely to develop after a decade or two of failure, a feeling of futility and frustration. I have been working in this cancer field for more than 20 years. You can feel my feelings. And another one is that believing nothing just because a so-called wise man said it is written down in books. Belief is generally held. Someone else believes it. Believe only what you yourself believe to be true. That's one of the drives that keep me still working in this field. Here is the outline of my presentation today. The new theory of cancer. Cancer is a functional tissue for wound healing. The evidence is to support the new theory. The role of the IBC in the new theory. The last one is the implications of this new cancer theory on IBC protocol. Excuse me. Now let's talk about the new theory of the cancer first. The current cancer theory holds that cancer is a genetic disease caused by the progressive accumulation of mutations in critical genes that control cells' growth and differentiation. Most mutations can be repaired by our system, but some of them escape and form a cancer. With the accumulating mutation theory, it is hard to explain the following phenomena. More than 50% of the SD rats will develop spontaneous tumors in 2 years, while it is less than 0.05% in humans at 2 years old. The doubling time of the SD rat's bone marrow stem cell is 1.49 days, and the human mesenchymal stem cell is 1.64 days. The division rate of the SD rat is similar to humans, indicating the chances for the SD rat's mutations should be similar to humans. Why does the rat have more tumor than a human at 2 years old?
So, silly question, what's SD stand for?
Sprague Dolly. Sprague Dolly. Sprague Dolly rat.
Sprague Dolly rats. Bacteria are in the human colon all the time. If the mutation causes colon cancer, why not on the bacteria in the same environment? The cancer incidence rates increases along with the age until 85 years old. After that, it declines. If the mutation caused the cancer, why does the cancer incidence lower after 85 years old? If the cancer is not a mistake by a mutation to genes, we may need to look at it from another angle. Let's look at some facts first. Cancer only occurs in organisms with wound healing capacities. For example, a pine tree, it can live up to thousands of years. There's no wound healing, there's no cancer. You just think about the pine tree, exposure to a sunshine, a UV, for so long, every day. But there's no mutation. Or we say it this way, there's no single or mutations or copy mistakes cause a tumor in a tree, although the cells divided every day. Another example, protozoa. They can live million years, but there's no wound healing, no cancer happen on them. For short-lived insects, for example, the Drosophila, they can only live up to 30 days, but they have a wound healing and they have cancer. Another example, the long-lived insects, queen termite, they can live up to 50 years. No cancer, no wound healing. For the mammal, they have the wound healing and they have cancer. The wounds are involved in pre-cancer diseases. For example, the smoking causes the chronic inflammation of the lungs and is a risk factor for lung cancer. Inflammation of the liver, colon, and the prostate increases the risk of the cancer developing in those tissues. Cervical erosion is a risk factor for cervical cancer. Ulcers are the risk factors for the gastric cancer. Oncogenes are involved in growth and the wound healing. The oncogene is originally defined as a gene that contributes to the production of a cancer. However, oncogenes are expressed not only in cancer, but also in pregnancy, embryonic development, wound healing, and a growth factor synthesis. Oncogenes also have broad anti-apoptotic functions. All those commonalities between the wound healing and the cancer above point to a mechanism evolved by the nature and lead us to a new cancer theory. Cancer is a functional tissue for wound healing. When a wound occurs, the body starts the wound healing mechanism. Many repair cells are used for the replacing the dead cells. However, if the wound is persistent, local repair cells may be exhausted. Then the oncogenes will be activated to secrete more growth factors and the cytokines. The factors will recruit more repair cells to the wound site, cause the differentiation and repair the wound tissue. Mostly, the wound is healed. Oncogenes are deactivated, and the metabolism goes back to normal. However, if the wound is still persistent, like a chronic inflammation, chronic infections, more cancer cells are divided rapidly to secrete more growth and repair factors for wound healing. Then a clinical cancer forms. After the wound is healed, the cancer cells can be redifferentiated or apoptotic. Then the clinical cancer mass will be gone. But if a small cancer cannot heal the wound, the tumor mass will continue to grow until the wound is healed or the whole system is exhausted. Now we can explain those phenomena that are hard to explain by mutation theory. Why the rats have more tumors than the human at 2 years old. The rat lifespan is 2 to 3 years. while a human lifespan is more than 100 years. Many apoptotic and malfunctioning cells exist in the old age body of the rats at 2 years, turning the oncogenes on frequently to heal the wound, while those old dead cells are not in the 2-year-old human, so that the cancer wound healing mechanism needn't be activated for that reason. Why does the colon develop cancer but not the bacteria in the same environment? Because the cancer program exists in human cells for wound healing, while the bacteria have no wound healing mechanisms and no such program, no cancer. Why does the cancer incidence lower after 85 years old? The functions of all cells will decrease when they are getting old. Cancerization is a function of a human cell, and it decreases along with other functions after the cell is getting old. Age 85 probably is the turning point that cells were really getting old in average. The old cells have less power